So now we know that antibiotics are required to kill bacteria, and we said that when antibiotics were first discovered by Alexander Fleming in medical world, it was a very good hope and real hope that antibiotics will uh, help us to eradicate bacterial infections. We can give antibiotics for every ill subject, and infections, bacterial infections as minimum, will not be possible anymore. It will be very easy to cure any episode of bacterial disease. But of course, you all know and you understand that that miracle didn't happen, and still in nowadays we have very high rate of bacterial infections, and still the same diseases are registered and uh, present in medical practice. Why it is happening so? So actually, very soon after penicillin was introduced into medical practice, it became known that actually it's not so easy, and bacteria, as any alive organism, can adapt to changing environmental factors. It can undergo evolution, and of course, with the time, it became known that some bacteria became uh, not susceptible to penicillin, they became resistant. And nowadays, the major problem of infectious diseases of medical care is antimicrobial resistance. So, what is it? Antimicrobial resistance is the ability of a microorganism to withstand the effect of an antimicrobial drug. So antimicrobial resistance, it is ability of a microbe to withstand activity of your therapy of your drug. And that is a problem because actually nowadays a lot of different microbes became resistant to multiple antibiotics, and we still have infectious diseases. They are even more aggressive than there were previous decades and previous century, but still infectious diseases happening daily. And that ability of microorganism actually can be of two types. It can be intrinsic, when innate ability of a microorganism species to resist activity of particular antimicrobial agent through its inherent structural or functional characteristics, which allow tolerate of particular drug or antimicrobial class. Complex definition, but in common if that ability is present in the whole species of bacteria or the whole species of microorganism, and every bacteria of the species can withstand activity or tolerate particular drug, then that resistance is called as intrinsic. So it is innate ability of the whole species of microorganism. It is ability to resist activity of a particular antimicrobial agent, and it means that you cannot use that precise particular antibiotic if infection is caused by that species of microorganism. And of course, in nowadays, intrinsic resistance or innate resistance or natural resistance, it is described for every bacterial species, and we have common charts where all data is present which antibiotics should not be used against uh, known species of bacteria. And of course, it is more easy if you already know which antibiotic you should not use. Just don't prescribe that drug, and you will have no problems with your therapy. But the major problem in that case is acquired resistance. Acquired resistance is a resistance when a particular microorganism obtains the ability to resist the activity of a particular antimicrobial agent to which it was previously susceptible. It means that naturally bacteria, that species of bacteria, should be susceptible to a drug. But when you will prescribe the drug, it will appear that drug is not active. And actually that precise bacteria, not the whole species of bacteria, that precise strain which is present in your subject became resistant. And you thought that the drug will be active, but actually when you prescribe the drug, it's appeared the drug is not active. and you need to change your treatment very fast as soon as possible to make your subject to give a chance for your subject to cure the current episode of infection. And acquired resistance cannot be predicted. We cannot know which mutation occurred, which gene appeared in that precise bacteria, and which drug will be efficient, will not. And of course, acquired resistance should be only studied. If intrinsic resistance can be predicted by looking at the chart, acquired resistance, it is always uh, something accidental. And to evaluate activity of a drug, you always need to perform some testing with your precise particular strain to be sure that some drugs or drug you prescribe is still active in that microorganism. So that phenomenon of antimicrobial resistance is a huge problem in medical care because any bacteria can become resistant to any drug. And it's not possible to guess or to predict, to make some model to say when and which bacteria will become resistant to which drug. So that problem, of course, should be solved, and we have a number of tests to evaluate resistance we will describe a little later. But the very first question when resistance appeared and became known was how it is possible that microorganisms which a day ago was susceptible became resistant to that antibiotic. And nowadays we have multiple different mechanisms described how bacteria can resist activity of antibiotic. We will list main of them, 6 mechanisms of antimicrobial resistance. First, and the most popular, it is uh, enzymatic modification or degradation of antimicrobial agent. Bacteria can produce a lot of and secrete a lot of enzymes which can change the structure of antibiotic. It will lose efficacy. Or some enzymes even can destroy antibiotic. And those enzymes are released into environment, and bacteria is surrounded by those enzymes. And when antibiotic will try to get closer to bacteria, and we already know that antibiotic should affect some precise organelle or structure of bacteria. It should get into direct contact. So when antibiotic will try to get to bacteria, close to bacteria, it will be inactivated or even destroyed by those enzymes. And then nowadays we have huge number of different enzymes, mainly um, it is different types of beta-lactamases which can inactivate beta-lactam antibiotics such as penicillin, but other as well I described. And those drugs, those enzymes not allow our antibiotics to get closer to bacteria and have an effect. And of course, in that case, efficacy of a drug will be lost. Second mechanism, it is changes in permeability of bacterial cell wall which make or restricts access of antimicrobial drug to bacteria. Means that cell wall became more rigid, pores became smaller, polarity may be changed, some other compounds are changed, and drug cannot pass into the bacterial cell. And again, we know that we have a lot of different mechanisms of activity, but mainly they all require antibiotic to pass into bacterial cell. They should get to ribosome, they should get to some enzymes, they should block synthesis of DNA, for example. But to have that effect, antibiotic needs to pass into bacterial cell. And if cell wall became very rigid or narrow or modified and antibiotic cannot pass into bacterial cell, of course it will have no effect. It will accumulate in sufficient amount around the bacteria, but it cannot pass into the bacteria. And of course, efficacy of the drug will be lost. Third mechanism is active efflux of antibiotic or antimicrobial drug from microbial cell into environment. And again, mechanism is realized by multiple efflux pumps. It is a kind of, we call it a pump, because antibiotic can pass into bacterial cell, but before it will get to a target, before it will fix to ribosome or block some other enzyme, it will be very fast excreted outside the cell. Of course, that process requires some energy and bacteria will lose some energy. But again, antibiotic will present in sufficient amount in tissues, but it cannot be holded in the bacteria. It will have no time to fix to a target organelle. It will be always released outside. And of course, antibiotic again will lose an effect and it cannot realize its potential. Bacteria will not be affected by the drug. So one more mechanism, it is a modification of a target for antibiotic. It means that, for example, antibiotic need to fix some enzyme. It is shown as a target several white lines, spherical lines, and blue center. So, of course, antibiotic is a chemical structure. It's similar to effect of receptor. Antibiotic need to recognize unique structure inside or on the surface of some organelle or enzyme, and it can fix only to known locus of that organelle or that enzyme. And of course, if structure of the target will be changed, antibiotic will not recognize. It's kind of a key in a door. If door lock is fine, key will open it. But if you will modify something, you will cut something or put inside something, key will not match and you cannot open the door. Similarly here, if structure, three-dimensional or chemical structure, polarity of a target is changed, antibiotic will not recognize that structure. And of course, that organelle, that enzyme will continue to perform its function, but it will not be blocked by antibiotic. And it will accumulate in sufficient amount even within a cell, but it will have no effect on modified structures and bacteria will remain viable. So one more mechanism is acquisition of alternative metabolic pathways to those inhibited by a drug. It means again that bacteria have not single enzyme and it have some alternative pathways for some enzymes and some organelles can take functions on affected organelles if it is required. And if, for example, antibiotic will get close to the cell, it will diffuse inside the cell and it will accumulate within the cell in sufficient amount. Target is not changed, it will block the target and initially some pathway will be blocked. For example, some production of energy by bacteria is blocked. But bacteria can activate alternative pathways. It can activate another enzyme with similar activity but absolutely different structure. And those enzymes, alternative enzymes, they will take the function of affected one. An antibiotic will work, it will block structures, it will perform its function. But bacteria will survive because it have another way, another enzymes which cannot be blocked by current antibiotics. And the last mechanism we will describe today, it is an overproduction of target enzyme or antimicrobial target. Again, we will prescribe antibiotic in some amount, and that amount is usually sufficient to suppress the life of bacteria, because we know that every bacteria have some precise number of enzymes that have some dose which can kill bacteria, dose of antibiotic. And when we give that antibiotic, for example, again, no other mechanisms are present. Antibiotic will diffuse in the cell, it will block enzyme is affected, but bacteria can initiate overproduction of that enzyme. It will be produced in so high amount that dose of antibiotic we gave will not be sufficient to block every target. And bacteria, again, it will lose some number of targets, some number of enzymes. But in general, amount of newly appeared targets will be sufficient to continue life and bacteria will not be seriously affected by the drug. So very often in clinical and routine practice, we see cases when bacteria have multiple mechanisms. It can have production of enzyme and active efflux, for example. Some antibiotics will be inactivated by enzymes. Those who will remain active and will diffuse in the cell will be excreted by efflux pumps. We can have modification of a target with overproduction of targets, and multiple combinations, of course, are possible. And one of the purpose of pharmaceutical companies nowadays is not development of new antibiotics. It is production and design of substances which can block mechanisms of resistance. For example, inhibitors of enzymes which can block enzymes, and those enzymes will not be able to destroy antibiotics. And it is again new direction, and nowadays we have multiple different combinations of antibiotics plus some substance which can increase or restore activity of that antibiotic if some mechanism is present in precise bacteria. So, of course, now when we said about dose, amount, we need to explain that actually antibiotics are a very complex group of drugs, and they should be prescribed in precise dose. And actually, antibiotics can be active, can be inactive. And to evaluate activity of antibiotic, we should identify MIC, or minimum inhibitory concentration. It is always the key parameter we will look for in any test. And minimum inhibitory concentration is the lowest concentration of antimicrobial drug that will inhibit the visible growth of microorganisms following overnight incubation. And in routine practice nowadays, very often and usually MIC is reported as a mass in some volume, of course. Any concentration is measured as mass of some substance in a volume of solution. And MIC is reported in microgram per milliliter or milligram per liter. In some old antibiotics, especially penicillins, for example, you can find IE, international units. But nowadays, that type of measurement is usually not used, and any test is targeted to identify MIC, minimal inhibitory concentration, lowest concentration of an antimicrobial drug that will inhibit the visible growth of a microorganism following overnight incubation. And why do we need to find MIC? Why do we need to look for concentration? As far as any drug is dosed in tablets or pills or milliliters, why here we need to find concentration, and which concentration is it? So please remember that antibiotic, first of all, it is a toxin. It is very toxic substance. It is real danger. And it is even named as antibiotic, against life. And that definition refers not only to bacteria, that definition refers to human and animals as well. Because actually, antibiotic, if it will be overdosed, it can seriously harm our body. And we have huge number of different side effects and adverse effects in the case of antibiotics. If you will take any instruction, any reference for antibiotic, it is always huge list of adverse events, a lot of. And we have cardiotoxicity, we have hepatotoxicity, renal toxicity, neural toxicity, multiple different types of toxicity described for antibiotics. And it is very important to give antibiotics in correct dose, because if you will overdose the drug, you can harm your subject more seriously than it will than infection will do. Infection that you're trying to treat. And antibiotics are very complex. Again, I will say it every time during any discussion that antibiotic is very serious group of drugs. It is very complex and very difficult to deal with antibiotics. And dosage arrangement for antibiotics should be absolutely correct, because you always will have very high chance to harm your subject more seriously with drugs than bacteria will do. So again, why do we need to find concentration? As any poison, antibiotic have safe interval in which it will not seriously harm the subject. and you can identify multiple different definitions and explanations. But as far as you are medical students and you all again going to be doctors, I will use medical definitions. And antibiotics have two important concentrations. It is minimum effective concentration, or MEC. It is minimal amount of a drug which will provide your subject with therapeutic effect, and minimum toxic concentration. It is minimal amount of antibiotic which will induce adverse event or side effect in your subject. And interval between MEC and MTC is known as therapeutic window. That interval when drug already have positive therapeutic effect but still do not have any side effects, that window is the perfect interval for therapy. If that window is large, then your chances of successful therapy will be much higher. It's easier to dose drug and more difficult to overdose it. But if that window is very narrow, if it is small, then of course it will be difficult to dose the drug. You need to perform therapeutic monitoring, you need to watch for adverse events for more detailed manner. And of course, we all know that humans are quite stable beings. Our properties with generations are not changing significantly. We have multiple mechanisms for control of our properties, for our evolution. We have very strict mechanisms of mutation control, and usually property of a human not changing significantly between generations and within one generation. So minimum toxic concentration is good, known, described, it is identified already, and again, you all can find toxic concentrations of different drugs and specific literature and specific references, and they are not changing significantly. But that minimum effective concentration refers to susceptibility of bacteria. And of course, you all already know that bacteria usually multiplies twice an hour, and every hour we have two new generations of bacteria, and every can mutate. Mutation is one of the main mechanisms of bacterial evolution, and mutation is a very common and frequent thing in microorganisms. And of course, every new generation will have very high chance to have different susceptibility to antibiotic. And that's why we need to monitor activity of the drugs. We need to identify that effective concentration which can kill current bacteria. And if that minimal effective concentration is lower than toxic one, then antibiotic can be safely prescribed to the subject, because the drug will have therapeutic effect but will not have toxic effect. But very often, minimal effective concentration can be higher than minimal toxic concentration. It means that if you will provide the drug, if you will prescribe the drug in minimum effective concentration, already that amount of drug is sufficient to provide your subject with a lot of different side effects. And of course, in that case, it is important to know how higher is effective concentration than toxic one. That's why we need to identify concentration, as any poison activity of antibiotics is measured in concentration. And our task is always to find that minimal inhibitory concentration, actually it is an equivalent of minimum effective concentration. And to compare it with the toxic, we should know, is it safe to give that antibiotic to the subject or no? And if it is so, then we can prescribe the drug. If our toxic concentration is much lower than effective one, then of course we need to look for another therapy option, to another drug or other methods to deal with that current episode of infection. So according to susceptibility of bacteria to a drug, we have three important groups of microorganisms: susceptible, intermediate, and resistant. Again, you can identify and you can find in literature a lot of multiple definitions, but you should understand, just if you will understand, it will be easier for you. Susceptible bacteria is bacteria for which minimal inhibitory concentration is much lower than toxic concentration. Means that if you will give the drug a minimal dose, it will kill bacteria, but it will not harm your subject. And actually, susceptible bacteria, it is a bacterial strain which is inhibited in vitro, in test tube, and some other tests by a concentration of a drug that is associated with the high likelihood of therapeutic success. Bacterial strain is inhibited in vitro by a concentration of drug that is associated with the high likelihood of therapeutic success. Means that it is highly possible the drug will not harm human but will kill bacteria. Intermediate strain, or abbreviated as I, again in literature is to just to save the space and to make it shorter. Susceptible strains are abbreviated till S, capital S letter, intermediate I, and resistant R. So intermediate strain, it is a bacterial strain which is inhibited in vitro by a concentration of a drug which is associated with uncertain therapeutic effect. It means that minimum toxic concentration is very close to minimum effective. They're very close. And you cannot know which effect of a drug will prevail, therapeutic or side effect. And in that case, usually intermediate bacteria require prescription of that drug in higher dose to be sure that bacteria will be inhibited, or prescription of a drug in combination with other. But of course, if you have an alternative, if you have some drug which is more active, then you should prefer drug to which bacteria is susceptible. But in some cases, when we don't have other options, or we have several drugs which are intermediate active, we can prescribe combination of the drugs. And resistant bacteria, or R bacteria, it is a bacterial strain which is inhibited in vitro by a concentration of a drug that is associated with a high likelihood of therapeutic failure. Means that minimal inhibitory, minimal effective concentration is higher, much higher than minimal toxic concentration. And if you prescribe that drug, you should understand that you will have very low chance to suppress bacteria, but the drug will show a lot of different side effects. And of course, resistant bacteria should not be treated with antibiotic, with current antibiotic. If you identify that your microbe is resistant to certain antibiotic, choose another one. Again, usually we perform testing for multiple antibiotics, of course not for single one. And always you will have a result for several drugs. You need to choose antibiotics with best activity, those which are susceptible or is minimum intermediate. If your drug is resistant, then of course it is very difficult task for a doctor. You will need to look for other options, other alternatives, or choose some combinations which will be more efficient. And again, it is a risk, of course, it is a risk, and it will be your duty to describe why you gave antibiotic if you get that result and bacteria resistant. What was the main purpose? Why didn't you choose any other alternatives? And always to protect you and to save you in any case and case history and ambulance card, always record indications why you gave antibiotic, explain why it was so important, and explain why you choose that precise drug. It will be more safe for you, because nowadays a lot of problems with some insurance companies and with the government, with laws, multiple companies checking prescriptions of a doctor. They're trying to look for some mistakes. Again, you know that it is a common thing when patients trying to sue doctors to get some money. And again, because of antibiotics are very toxic, they can have a lot of side effects. It's better to record a reason why you prescribe the drug, how long should subject take the drug, and which dose did you choose and why. It will be more easy for you in the future to explain why it was so important to prescribe a toxic and potentially dangerous drug. So, how to find that MIC, or minimal inhibitory concentration? And nowadays we have multiple different tests a lot of tests, a lot of modifications, but to make it a little easier, we will explain and we will describe one of the very first tests. It is a serial dilution test or broad dilution test or method. To perform the test, again, you need to take several test tubes with 1 milliliter of nutrient broth suitable for cultivation of bacteria. Again, the main task for you is to make multiple tubes with different concentrations. And of course, you need to compare, then you need to inoculate bacteria in every test tube, incubate for 37 degrees for 24 hours as minimum at night. And then you will compare results, you will check your results. You will check if bacteria in that current test tube is growing or not. And minimal concentration, which will give you visible inhibition of the growth, will be MIC. So first test tube, when bacteria is growing, bacteria is not inhibited. So the concentration is not sufficient. Previous one will be MIC. But the very first problem occurred. Which concentrations to study? Which amount of drug to take? Because actually, if I will take, for example, 10, 9 milligrams per liter, 8, 7, 6, 5, 3, 2, 1, will perform the test, I will get some result. For example, 3 milligrams per liter is MIC, it is fine. But another laboratory, some Indian doctor, will say, 10 test tubes, it is expensive, it is time consuming, I will take 5. I will take 10, 8, 6, 4, and 2. It will be sufficient. And the doctor will get, for example, 2 milligrams per liter. I have 3 with the same strain, the doctor will get another number. And in some Australia, doctor will say, no, guys, it's too much. 1 milligram per milliliter difference is too much. We need to take 0.5. And he will make 10, 9.5, 9, 8.5, and in general, he will perform 20 tests, but he will get some 2.5. And in general, we 3 doctors studied the same bacterial strains, but we all get different results. Some 4, 2.5, and 3. Why? Because that concentration used by us was different, and range was different, and method was different. Second problem, how many bacteria to add? Some people will add 1 loop, some will take 1 milliliter, some will take 1 microliter, and of course, number of bacteria is very important because concentration will be changed of bacteria. And the same amount in test tube on 1 cell will have much more good effect than in 100 of cells. So it was decided, initially it was decided that any test in any country, regardless of geographic location, regardless of profile of hospital, laboratory, every test should be performed in absolutely precise manner. Every laboratory should use every step the same. And if every doctor will perform the step according to one common algorithm, then those results can be compared. Any laboratory in the world can compare their results with some international publication, with some other data. And nowadays it was decided that it was initially, of course, it was identified by multiple experiments, then modern antibiotics can be used in maximal concentration of 512 milligrams per liter or micrograms per milliliter to 512. And that concentration is maximum possible, maximum required for any drug. So to perform tests, of course, you need to take highest concentration of a drug. So every test tube should contain the same amount of antibiotic and nutrient broth. So in classic manner, you need to prepare required amount of test tubes and add 1 milliliter of broth in every test tube. Sterile with no antibiotic, no bacteria, nothing, only broth. And in the first test tube, you will add 124 1 milliliter of antimicrobial solution with concentration 124 milligrams per liter. Of course, you will dilute it in twice, 1 milliliter of empty broth plus 1 milliliter of your solution, concentration will decrease in twice. And in first test tube, you will have 512 micrograms per milliliter. It is the highest required concentration. Then you need to shake to mix the test tube and transfer 1 milliliter from first tube to second one. Again, you will dilute your concentration in twice. And now you have 256. And you will perform that activity for every test tube. And in general, you will have, we call it serial dilution because we will perform a serial of dilutions, and in every further test tube, concentration of the drug will decrease in twice. And any laboratory nowadays using the same numbers, 128, 64, 32, 16, 8, and etc. Of course, previous antibiotics, one of the first antibiotics, were less effective, and concentrations for them were pretty high. And nowadays, modern drugs can be active in concentration lower than 1 microgram per milliliter. And we have 0.5, 0.25, 0.125, and concentrations much lower than 1 microliter. Again, for every drug, we have described concentrations. We have a chart which range of concentration to study for every antibiotic. And of course, if you need to perform some tests, you need just to take the chart and take a look which concentrations should you prepare. But they all originate from initial concentration 512. They can be diluted in twice, but they're always equal. And of course, you need to inoculate known amount of bacteria, known concentration of bacteria in every test tube. We have standard of density, which is measured in McFarland's. We have devices which can measure the density in classes. You will perform those tests and you will see how it's working. But again, you need to prepare initial one common solution with the density 0.5 by McFarland and inoculate fixed amount of that solution in every test tube. And in that way, you always will have the same volume of studied mixture, the same amount of antibiotic, the same amount of bacteria, and the results will be comparable with common standards. And evaluation of the result, again, can be performed visually. You should see MIC, it is inhibition of visual growth. And as you can see on that picture, please take a look on the slide. You can see that first 3 test tubes are absolutely transparent, simple broth without any visible growth. But 16 micrograms per milliliter, that test tube giving you turbidity and visible density. And that test tube bacteria is growing. So 16 is not enough to inhibit the growth. So 32 micrograms per milliliter in that slide is MIC. So first to the last test tube, when bacteria is still not growing, it is the test tube with MIC, minimal amount of a drug. And of course, the test is easy to evaluate, easy to perform. Just perform few activities, filled in those test tubes, incubate, and you have a result. But you get that number. What to do with it? We get that it is 32. And so, of course, we all need to use that result for routine practice. We should know, is that concentration is sufficient to inhibit the growth and not to harm our subject or not? Of course, no one will ask you to remember all concentrations for every drug. And moreover, with time, with years, those concentrations can be different because bacteria is adapting, newly drug appear, and actually, nowadays we have 2 major standards, 2 organizations or 2 institutions which deal with evaluation of antimicrobial activity. It is a CLSI, Clinical and Laboratory Standards Institute. It is an organization located in USA. And it is a EUCAST, it is European Committee on Antimicrobial Susceptibility Testing. It is, as it is obvious, located in Europe. And 2 organizations, they perform regular and constant monitoring of activity of multiple antibiotics. And annually they publishing guidelines of antimicrobial testing. And of course, no one will try even to remember every numbers, but you can open guidelines and you will see charts for every bacteria or bacterial group. Again, two standards can be a little different, and in our continent, we're trying to use UCAS because European Committee and European bacteria, of course, are more closer to us, and moreover, UCAS is free. The guidelines are published in website. You can easily take a look on them. They're free of charge. Anyone can open and read it. CLSI is more old organization, more experienced organization, but their guidelines usually are you need to pay to buy that book or to buy access to the site. And usually only companies and some organizations using CLSI guidelines. If it is some small, simple experiment, of course, it's more easy to use UCAS guidelines. They are similar, but in some cases they can be different, as you can see on that slide. So it is a small textbook, very small text. It's looking like a copybook. And when you will open that book, it will be some common information, how to perform tests, some common guidelines. And after that, you will identify, you will find charts for different groups or different species of bacteria. For example, some enterobacteria, Staphylococcus, Streptococcus, and when you will open it, will be a list of antibiotics, as you can see on the slide, in the left part of the slide, list of antibiotics, and three categories of bacteria, S, I, R, susceptible, intermediate, and resistant, and some number. It is a concentration given in milligram per liter, equivalent of microgram per milliliter. And you can see that it is some, for example, ampicillin, and concentration is equal to 16 milligrams per liter. Then according to CLSI, that bacteria is intermediate. According to UCAS guidelines, that bacteria is resistant. And you should just compare your result with the result provided in the chart. And you will see, can you use that antibiotic or not? And of course, when you will have a report from your laboratory doctor, it will be usually marked which drug was tested and which result was obtained, resistant or susceptible bacteria. Sometimes doctor, if it is required or if it is a common practice, doctor can write down even number. For example, some ampicillin 32 resistant. And when you will get that information, of course, the very first task for you is to adjust antimicrobial therapy. You will see that antibiotic which you prescribe is effective in current case or not. If it is effective, you can continue with the drug. If your antibiotics fall into category of ineffective, then of course you should switch therapy and prescribe correct drug. So again, the only task of that method, of any method of antimicrobial susceptibility testing, is to find MIC. And laboratory doctor, will provide you with information about MIC of different drugs. And you can compare, or laboratory doctor will compare result of current test with common standards and give you information, is the drug effective or not, if bacteria is resistant or susceptible. Of course, if you will see in report that drug is ineffective or bacteria is resistant, don't use the drug. If it is already prescribed, switch it and explain, make a record that according to obtained result, according to received result, antibiotics prescribed initially was or is ineffective. That's why I will choose another therapy option. I will prescribe drug which is effective and bacteria susceptible. Just give that explanation and you will make your life easier. You will release a lot of questions which can originate from checkers, from insurance companies and other. Of course, the test was one of the very first, but nowadays it is replaced, broad dilution test, because it consumes a lot of reagents, a lot of antibiotics. You need to prepare 1 milliliter of every concentration for every bacteria. In general, some, for example, 10 tests can require some liters of antibiotics. And some drugs are really expensive and substances are really expensive. And of course, common idea of current method is to decrease price of the drug, decrease consumption of antibiotics for those testings, and broad dilution test is replaced with the broad micro dilution test. It is a common 9612 plastic plate. You can add microliters of a drug in every in every well, usually 100 of microliters or 50 microliters. And of course, principle is absolutely the same. You need to add drug in different concentrations, and it is already prepared raw for you. Add bacteria in fixed amount in every plate, incubate in every well, incubate, and of course, as far as amount of solution is much lower, result will be visible more faster. And usually it is not a day, it is night incubation for 8 or 12 hours. And the result is the same as you can see. For example, row A, wells from 1 to 4 are clean, empty, and no visible growth is acute. In A5 well, you can see that some already inclusion is present, and means that the concentration is already not sufficient. And in the last, in the 12 row and 11 row, it is absolutely obvious that bacteria is growing here. So test of microdilution consumes less amount of antibiotics. It saving space in incubators because one small plate, it is some about maybe 20 or 30 centimeters in length and few centimeters in height. Of course, it consumes a lot of space. In the same incubator, you can put about 10 microtiter wells instead of one set of test tubes if you will perform simple broad dilution. So another method which is still kind of popular, and you will perform again the test in laboratory classes in practice, it is a disk diffusion test or disk diffusion method, abbreviated as DDM or DDT. Principle is very similar, but here, as you can see, we're using petri dish, one petri dish instead of test tubes, and of course it is agar. So the principle of the test, you need to take filter discs made of synthetic cellulose or some paper, and every disc have absolutely the same size. And antibiotics initially are added on those discs by manufacturers, of course, not you will do it. Every disc have fixed amount of antibiotic and have fixed diameter. You need to take petri dish, inoculate with your studied bacteria, and add discs containing different antibiotics on one petri dish. Incubate, and as the result, of course, in empty agar, bacteria will grow, and antibiotic from the disc will diffuse in the agar. Agar is porous material that have a lot of channels inside, and antibiotic from disc will diffuse in the agar. But with the distance far from disc, concentration of antibiotic will decrease. Around the disc, concentration will be highest, and some amount will diffuse further, and of course, concentration of drug will decrease with the distance from the disc. And bacteria will grow on agar, and it will grow only in those areas where antibiotic is present in insufficient amount. But as you can see, it is marked with a yellow arrow, and it is known that phenomenon is known as zone of growth inhibition. In some space and some distance, concentration of antibiotic will be sufficient to suppress the growth of bacteria. So in that zone, it is obvious, you can see the spherical area around the disc, bacteria is not growing. But in some point, bacteria appear. So in that precise point of agar, concentration of antibiotic is sufficient to suppress the growth. Here in that point, you have your MIC. How to measure? A lot of students will ask how to measure. We can take scale, we can measure distance, we'll get results in millimeters, but we need to have concentration. And of course, we cannot report result in millimeters. MIC is 15 millimeters, it's not correct. And of course, the perfect and the only possible way to find that concentration here and that precise point of agar is to cut that zone of agar, dissolve, and by common chemicals method identify how many drug is present here, usually by titration. But of course, in that manner, method became extremely expensive, time consuming, and requires a lot of equipment. So again, it was decided that every test should be performed in precise standard. And any company producing discs, any laboratory performing tests, always will take disc of the same density, the same height, the same diameter, and concentration of antibiotic, amount of antibiotic here on the disc will be absolutely identical. Agar which is used for test is always the same. Concentration of the agar, composition of the agar is the same. And again, it should inoculate bacteria in that petri dish in the same density always, 0.5 microfarad. And in that case, every result you will get will be absolutely comparable. Means if you will take the same agar, if you will take the same disc with antibiotic, that antibiotic will always diffuse in the same amount, in the same distance. And it was done only once. Every millimeter of agar was taken and concentration of different antibiotics was measured. And nowadays it is known that if you will perform test according to common standard, you always will have known concentration and fixed distance. And again, we have charts how to recalculate millimeters into concentration into milligrams per liter. It is known, for example, if drug diffused in 1 millimeters in your test, in that point you will have concentration of some 256. And all you need to do is to measure distance of that zone growth inhibition and compare your result with the chart. And you will know that if it is some 10 millimeters, then it is equal to some known concentration. Again, those charts are available for every antibiotics. And in that manner, test became more easier. All you need to do is just to measure zone of growth inhibition, and that's all. You have a result. You don't need to perform any titration or other studies. Test is fast and precise. But we have one penalty a little later. One more thing which was noticed in that test. We said that antibiotics can have summarized effect when one antibiotic increases in activity of another antibiotic. It's not really possible to find that synergy in broad because we always have only one antibiotic and one test tube. But in the case of this diffusion test on one petri dish, we have multiple antibiotics. And sometimes it is possible to identify that synergy. You can see that, for example, ceftriaxone have stable zone of growth inhibition around the disc, but when two discs are put together, amoxicillin and ceftriaxone, these antibiotics from disc diffusing one towards other, and in some place they will cross, they will accumulate both, and you can see that activity of ceftriaxone is increased when ampicillin or amoxicillin is present. And that synergy can be used in medical care. You can see that one drug is enhancing activity of another. And if you prescribe drugs in combination, you can decrease the dose of every drug, but efficacy will be still the same. So that is one more benefit of the method with the use of discs with antibiotics. But as we said, the major we have one penalty of that test. You can perform everything according to common standard. You can measure density of bacterial inoculum. Discs are always the same. Petri dishes are always the same. But one thing we cannot standardize, it is a scale. And scale which is used by for measurement of the zone is always different. And of course, you all know, and I think you already saw it, and you noticed that uh, differences can be very significant. If you will take different scales from different manufacturers, they can be significantly different. Some scale will show you 3 millimeters, another will show you 5 millimeters. In common practice in some school classes, maybe it's not very sufficient, not very important, not very significant. But if you need to evaluate activity of a drug, you need to measure distance in detailed manner, and every millimeter is extremely important, because in 1 millimeter concentration can be different significantly. Of course, it's not allowed to have some so significant error in a few millimeters, because data can be absolutely different. And because of that problem, some other problems are present as well. But that is one of the reasons why test is not very popular nowadays still, and other tests are preferred. Results depends on the scale, and we cannot check quality of a scale. So that's why nowadays we have epsilon tests or e-test. It is commercially available, again, synthetic paper strip with already pre-printed scale. And the major idea of the test, it's not simple scale. It is a strip with antibiotic in different concentrations. Not antibiotic will diffuse in the media, and you need to measure zone of growth inhibition. That strip already contains antibiotic in known concentrations. They are not common, they are not some 256, 128, 64, 32. More numbers are here, but it's not a problem because actually it can be compared anyway. It is precise. The test is always done by the same manufacturer, or scale is controlled by different manufacturers. They have one common standards, they're identical. And in that case, you will not have those errors due to different manufacturers of scales. All you need to do is you need to put that paper strip on the petri dish, inoculate it in the same manner, and incubate. And as a result, you will have that epsilon shape or tree shape zone of growth inhibition, and you don't need to measure anything. You need just to take a look in which point bacterial growth touch paper strip. And you can see in the slide it is some approximately 1.0. Bacteria is not growing around paper with higher concentration, but when concentration on that paper will decrease till MIC, bacteria will grow here, will touch that paper strip. And again, one of the benefits, you can apply several different paper strips with different antibiotics on one petri dish. For example, here in the slide you can see 6 paper strips on one petri dish, 1 bacteria, 6 antibiotics, and you will identify MIC in detailed manner. The same zone on growth inhibition, the same different concentrations of antibiotics, but we don't need to recalculate everything. It is already prepared result. And of course, the test is much more suitable, it's more easy, it's more precise, and the only penalty of that method is the price, because it's very difficult and it is expensive to make that paper strip. It is a few centimeters in length. Of course, it's difficult to control concentration till some 0.2 milligrams per liter. That's why those strips are expensive. And of course, if laboratory performing some hundreds tests annually, price of tests and price of those strips became very significant. And that's why that test is still rarely used. And nowadays, broad microdilution test is the gold standard of antimicrobial susceptibility testing. But anyway, some laboratories performing e-test as a parallel test or for some other purposes. It's faster and easier to perform. So, of course, and nowadays you have some other tests, but we will not talk about them because our time is limited. And just please remember that regardless of the method you're using, always your task is to identify MIC, minimal inhibitory concentration. And when you will identify MIC, you need to compare the data with the charts. You need to be sure that antibiotic will kill bacteria but will not harm the subject. That concentration, effective concentration, minimal effective or minimal inhibitory concentration is lower than toxic concentration for your subject. And if it is so, you can safely apply antibiotics. If it's not so, then it's better to choose other drug or use combinations. If some drugs have a synergy, then decrease the dose of antibiotic. But please be very careful because it is really difficult group of drugs and a lot of problems you can get when antibiotics are prescribed, especially if they are prescribed not according to indications. And one more thing, the last thing I want to talk and I want to tell for you, it is a common error, common mistake, and common false idea in doctors that antibiotics can be used for prevention of the disease. It's not so. Antibiotics are killers. They cannot prevent something. They can only kill something right now and right here. And if you have bacteria in subject, infection is already there, it is present. You need to prescribe antibiotic, it will kill that pathogen. But if microbe is not there, antibiotic cannot prevent its appearance. It will just circulate, it will look for some target, and the only target it will find if pathogen is not here. It is normal flora bacteria. And of course, it is absolutely the same target. And antibiotics and the major number of side effects are related to normal flora of GIT. Antibiotics will kill bacteria which is present in intestine. And incorrect and inappropriate use of antibiotics is one of the leading reasons of dysbacteriosis. We talked already about that condition when normal flora is affected and subjects suffering from serious consequences, and in severe cases, it can lead to ulcerative colitis and perforations and peritonitis and very serious complications. So antibiotics are only for treatment, only for therapy. If infection is here, treat. If infection is not there, do something else. Give a vaccine if it is a high chance that subject will be infected. Perform sterilization of instruments. You can clean surgically wound or some injuries, remove debris. But drugs will not prevent, and antibiotics will not prevent infection. And again, we have multiple groups. Antibiotics in classic definition are drugs against bacteria. They're not effective against fungus, against protozoa, against viruses. They only targeted against bacteria. And again, if subjects have viral infection or fungal infection, you will prescribe antibiotics. They will not affect pathogen, but they will affect normal flora. And if you need to give antibiotics, no other chances. Subject is already ill, serious infection, you need to give antibiotics for long time period or in high doses. Again, remember about dysbacteriosis. Give symbiotics, probiotics, eubiotics together with antibiotics. Because anyway, even if pathogenic bacteria is present, some amount of drug will affect normal flora. And anyway, subject will suffer from dysbacteriosis. So prescribe protective drugs to save flora of human because that flora have one more important property. It is our helper. Normal flora can help to resist pathogenic bacteria. And if you will prescribe antibiotics, protect normal flora, give some other biotics, any, Sin, U, Pro, L, any, but something to protect normal flora of the subject. And the last thing, it is important to remember that antibiotics are toxins and don't give them if they are unnecessary. If you need to treat infection, prescribe how long antibiotics should be prescribed, make a record, how long antibiotics should be prescribed, in which dose and which frequency, and when to check efficacy of the drug. Because usually drug should be effective in first 3 days. If you prescribe the drug, you see that no therapeutic effect, usually it means that that antibiotic not working. You need to recheck susceptibility because bacteria can already change its property and switch the drug, give another one. And always make a note in official papers, case history or some other, why you prescribe the drug, for how long, and when to check its efficacy, when to stop therapy. Because if you will go vacation or any other reasons you didn't attend the job, other doctors, they will not check your work. They will still continue with their prescription. Antibiotics can be prescribed for a long time period. So make a record for other doctors, for nurses, when to check efficacy and when to check if the drug is still required or not, or it can be stopped. And you will save your subject, you will save yourself, you will prevent a lot of problems, again, related with different instances, institutions such as insurance companies or other. So for current lecture, that's all. We will talk a little about antibiotics and you will perform a lot of practice in classes. See you next lectures. Bye bye.